let's jump in because we ended our lecture last time by talking about Louis Pasteur and his ingenious, eloquent, swan-necked flask experiments that disproved spontaneous generation. But remember, he didn't work alone. He needed the help of other researchers such as John Tyndall and Ferdinand Cohn to allow those who tried to repeat his experiments to understand that different kinds of broth require different sterilization times. So it was a team effort that really put the nail in the coffin of spontaneous generation. When we ended, we mentioned that the reason for the inability to repeat those experiments was that endospores are found in certain kinds of broth. Remember that endospores are the BA structures that can resist heat, desiccation, chemicals, UV, and other environmental stressors. So these endospores are, when the going gets tough, the tough form endospores. We want to now look at the contribution that Robert Koch made to, at the exact same time that Ferdinand Cohn was discovering the existence of endospores. That year, Robert Koch was working on this idea of attributing disease to a microorganism. Okay, so you can see how his work was on the back of that of John Tyndall, who said, wait, dust is associated with disease. Well, that's because microbes were in dust, right? And so Robert Koch was working on this idea of how do you show that a bacterium causes a disease? For example, how do you show that Bacillus anthracis causes anthrax? So let's take a moment to see if we can write down what are Koch's postulates. These are the postulates that allow for the connection to be made between a bacterium, for example, Bacillus anthracis, and the disease that it causes, anthrax. That is, you want to show, what would you do if you wanted to show that a bacterium was the cause of disease? Let's write down what we would want to show. So, number one, we would want to be able to show that the bacterium was present in all cases of the disease, right? So we would want to show that it shows up anytime that disease symptoms are there. So bacterium is present in all hosts with disease symptoms. But likewise, we would also want to show that it was absent from healthy hosts. So we can say, however, the bacterium is absent from healthy hosts. Now, secondly, we would want to be able to actually literally isolate the bacterium from the diseased host and grow it in pure culture. That is, grow it alone, away from any other microorganisms that may have come from that host. So we would want to be able to isolate the bacterium from the diseased host. So number three, we would want to be able to then take that isolated pure bacterium. So let's write this down. Let's say isolate the bacterium from the diseased host, and let's say in pure culture. And then we could say inoculate a healthy host with the bacterium and observe the disease. Now, just for good measure, we would then want to be able to re-isolate the bacterium. Now, these are considered Koch's postulates. They have been modified to express the link between viruses and the disease that they have can cause as well. However, we can also say that it's important to note that although these are very useful and they're still applied, it's very binary, and particularly this idea that bacteria are present only in those hosts that have the disease, because what we better understand now is the nebulous nature of that, because there are many bacteria that are opportunistic. That is to say, they may be present in a host, but they may only cause disease when given the right opportunity at the right time. For example, many of us are hosts naturally of a bacterium called Staphylococcus aureus. This is a pathogen. It can cause disease, but in those that harbor it naturally, it generally does not, unless there is some sort of state that that person is in which there may be immunocompromised, or there is some good entry port for the bacterium that it may cause disease under certain circumstances and not others. However, this gives us a little bit of an idea of the postulates that Koch put forward to allow him to understand Bacillus anthracis as the causative agent of anthrax. So with this in mind, I want to leave behind the idea of the historical contributions to disproving spontaneous generation and working on some of the foundations of the history of microbiology, and maybe we could call Koch the father of bacteriology. I want to now move away to a much broader picture of the applications and implications of microorganisms, knowing that they are absolutely pivotal to everything in our world. Microbiology certainly affects things that you never even knew were affecting you every single day. So I want to look a little bit at that, and I hope I can call many of you as examples into my coverage. Let's begin with essential environmental activities for microorganisms. And Leslie, you are going to be so jazzed because your passion for the environment is very much central to microbiology and much of what we will talk about with regards to microorganisms as the foundation of the food chain, but also their role in being important microbiota for the earth. So when we think, last time we mentioned about the human microbiome, the earth also has a microbiome, and if it's in balance, it allows for proper cycling and recycling of elements and nutrients. So this idea of biogeochemical cycling relies very heavily upon the microbial world. So microorganisms can cycle things like nitrogen, they can cycle things like oxygen, they even are involved in the manganese cycle. You may not have even known that that was a thing. So the sulfur cycle. So they are very important in converting atmospheric nitrogen into forms of nitrogen that can be used by plants and animals. They replenish atmospheric oxygen. In fact, you plant lovers didn't realize that in fact more than half of the contribution to oxygen is made by the microbial world. They decompose materials that no other organisms can degrade. So Eric, in a minute, I'm going to talk to you a little bit more about this, but this is this idea of biodegradation, the natural role that microorganisms play in decomposition, but then also the ways in which we can tap into their ability to biodegrade things and use that to our advantage. So more in a moment. Let's begin on the applications of microbiology. I want to talk to a couple of you who have passions about food, and I think Zeri, you were the #1 person to email me. You were so quick on the uptake. So Zeri is super stoked about food. Also, Jency loves to bake, and I think it was Kelsey who was out there who also said something about food and passion for food. So there are so many of you that have some sort of interest in nutrition, but also maybe didn't realize how much foundation the microbial world plays in food production and preservation, but not only that, in our own health, in consuming those preserved and produced products made with microorganisms. And I want to mention that historically we have known about fermentation for a very long time. In fact, it was in 2100 BC that the Egyptians first started to bake bread, so Jency will be interested in that. And it was in 1500 BC that they began to make beer. And so Danny, you had mentioned wine and beer tasting as being one of your passions. So this is something that has been very historically relevant, and I was even thinking about Borden mentioned her passion is regarding classic literature, and I was thinking about Dickens' references to ale, and particularly I was thinking about Great Expectations. I think there's actually a term called Dickensian, if I'm pronouncing that right, that just actually refers to his writing as writing about the kind of underbelly of society. And so there's a lot of references to fermentation, which is really cool. So historically, we see a lot of references to food production, microbial life within beer, wine, bread, soy sauce, yogurt, buttermilk, sour cream, cheese, pickles, and all of those fermented foods that we love the tastes and textures of their incredible contribution to our diet. But not only are they incredible with regards to allowing us to produce this food, they are also play a major role in food preservation. So Danny will recognize that wine is preserved. The alcohol that is a byproduct of fermentation serves a great role in preserving that food as well. And of course, Eric, I promised you a little bit of coverage on bioremediation or biodegradation. We utilize microorganisms in important economic application in trying to clean up our messes. So for example, many of you recall the Gulf oil spill and how incredibly deleterious that was to life. Obviously, ideally, we would prevent that kind of disaster. However, when it does happen, we can often apply bioremediation to clean up our messes. This is something that we're actually doing very close to home as well. If any of you spend time, like I take the ski team early season to do intervals out on the bike path, and the loop on the bike path actually has a lot of active phytoremediation. So this is actually a photograph that one of my students took out on the bike path where you can see all of the trees that they have planted there. This is enhancing bacterial microbial growth within the earth to allow us to remediate the oil that was deposited in the soil. So between 1928 and 1982, there was a massive amount of creosote, which is the railroad tie preserving oil that was deposited into the earth here. And engineers, catch this, they tried to use a dual drain system to actually flush the oil out, did not work, and so then they settled on phytoremediation, which has been incredibly effective. Phytoremediation can be used against things like to clean up things like oil, but it can also be used to clean up other things. This summer I did a fantastic project with a phenomenal undergraduate student. His name is Kyle. He was studying the use of phytoremediation to clean up uranium at the UMTRA site outside of Riverton. So some of you make a home there. And so cleaning up uranium from the soil is another application for bio and phytoremediation. We can also clean up things like TNT. There's actually something called the Sabre process that actually allows us to convert trinitrotoluene to triaminotoluene. And I know some of you said you had a passion for chemistry. Think that one through, because TAT is much less of a it's much less of a threat biologically, and certainly not explosive the way that TNT is. So lastly, in our list of economic applications, not to say that this list is very much a partial list, they can be used to produce things like antibiotics, amino acids, and insecticides. And there were a number of you that expressed passion for medicine, and certainly this is something where the forefront of science is based on antibiotic production, but even more so antibiotic resistance, and how we overcome that barrier. So in many ways, microorganisms are important to us there. It's worthy to say that the classical definition of antibiotic is actually a substance that is produced by one organism to kill off another or to inhibit its growth. It's classically thought about as biological warfare between microorganisms. So we've exploited that to a certain extent, isolating these chemicals from the bacteria that produce them and utilizing them in disease treatment. Now, more I promise to come on that. So let's turn our attention to genetic engineering. And I imagine that this evokes a great number of various imagery within your mind, right? Ranging from the hugest, largest, ripest tomato, perhaps all the way to thinking about treating leukemia with gene targeting. So this very much spans the board, and in fact, the definition for genetic engineering can be very broadly stated as introducing genes from one organism into another organism to give it new features and or abilities. So here are some ways in which we have very much utilized genetic engineering within medical sciences, but we could think about utilizing it right also in Eric, in bioremediation, by giving organisms new enzymes, right? The ability to, genes that encode for enzymes that would give them the ability to, say, degrade a recalcitrant compound. Recalcitrant means super, super hard to digest. So if we could give an organism genes that would allow it to degrade something that it wouldn't normally degrade, you can see applications, right, for bioremediation and environmental applications there as well. Some of the most famous applications, though, are medical, and the literally quintessential application is the one in which we think about using microorganisms to make insulin for us, to literally pump out hand over fist insulin and allow for the production of something that can treat diabetes. So this is something, again, that will super interest Zeri, right? So a lot of medical application. We can also use microorganisms to make blood clotting factors, enzymes that dissolve blood growth factors. So additionally, whenever we make a vaccine, we're in some way using a kind of genetic engineering. That is, we're replacing virulence genes within a pathogen with something that is a false threat. So let me explain that. What I mean by that is that take a virus that normally causes disease and remove the genes that cause it to make disease and replace them or get rid of them, and maybe allow it to have only a few genes that cause our body to respond to it, but not that cause sickness in us. So suddenly our body is mounting an immune response to this false threat that really won't cause disease, but yet our body has gained immunity to it. And that's the idea of the use of genetic engineering to make vaccines against various diseases. Bacteria can be used to genetically engineer plants, and the most, this is very, very common, right? We can genetically engineer corn, for example, to make its own insecticides. We can do this in the most common vector, that is the most common delivery agent for making these plants make their own insecticides or pesticides, is to take a bacterium that usually infects plants and gives them a tumor, but instead we genetically engineer this bacterium to have the genes that give the plant the ability to make the insecticide, or that encode for herbicide resistance, so that we can hit fields of plants with herbicides that would kill weeds, but then they won't kill the crops, right? We use a bacterium called Agrobacterium tumefaciens to do this, and we replace that tumor-inducing gene with the genes that, you know, give the plant the ability to make the insecticides, or that give the plant the ability to resist herbicides. So advances in gene therapy may allow widespread use of viruses to transfer genes to patients in order to treat genetic diseases. And this is where I want to talk to Daryl a little bit. Daryl actually has a nascent and budding interest for optometry. And as it turns out, one of the areas, gene therapy has had a lot of starts and stops, and it's been hard to get gene therapy up and running in an effective way, in many cases because when we're doing gene therapy, we're trying to give someone a gene that they lack in order to treat a disease, right, that's caused from lacking that gene. But a lot of times when we try to give them that gene, it causes them their immune system to look at that new product and say, whoa, foreign invader, and it wreaks havoc, right? So, however, Daryl, in optometry, we can see promising results of gene therapy to treat ocular disease. In fact, it's been one of the most trendy areas for treatment of disease with genetic engineering. And this particular press release, it's a public perspective article, Science Daily, but it talks about 2 journal articles in human gene therapy that looks at the idea of using gene therapy to preserve photoreceptor function and maybe to then treat retinal degradation or degeneration. Excellent. So a lot of positives here, but we also really need to consider some of the ethical, moral, I don't love the word moral, but some of the ethical sort of even political, honestly, policy type questions that surround genetic engineering. There are a lot of questions that come up when we talk about genetic engineering and cloning. And so this is something that I think is worth our time to just consider. What are some scientific, philosophical questions that surround genetic engineering? Together, we're going to consider some answers to this question regarding scientific, philosophical questions perhaps that surround genetic engineering. But I want you all to think about this question on your own as well. And that's why in the homework I am actually posing this question, and I want you to come up with questions that this evokes for you. And maybe they are some of the same questions that we will discuss, but perhaps there are others that you might want to contribute. I'm going to begin with one that I think is going to interest Lorraine, and I hope I'm saying your name correctly. This is something that is very poignant in agriculture, and this is the question of who owns genes. So the answer to this perhaps is more complicated than you might think, because right now the patent laws stand such that you can actually patent any gene that is not quote unquote naturally occurring, which I think is a super hard thing to define when we think about our lack of knowledge of what is out there with regards to bacterial biodiversity. We may not know what genes are out there, but many of you are familiar with the fact that companies can own genes. So there's actually corporate ownership by, for example, Monsanto. And some of you are familiar with something that we call BT corn and BT cotton. These are plants that have in them the genes that come from a bacterium called Bacillus thuringiensis. So first name of the bacterium, first and last name of the bacterium is where the BT derives. So these have genes that come from a bacterium that allow them to produce their own pesticides. And when a farmer plants BT corn or BT cotton, that farmer does not own the corn and cotton. The corporation that sells those seeds does. And so this question of who owns genes is a very, 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 very nuanced one, a very interesting one. The other question that is born from this question is the idea that when we plant BT corn and BT cotton, we have these fields upon fields upon fields of genetically identical plants. That is, we are growing a monoculture. So in monocultures, what we lack is biodiversity. So I'm just going to jot a note that just says lack of biodiversity in monocultures. Now, this can be devastating because typically in any normal population, there's at least 10% biodiversity, and that protects a population from disease. So one of the great quintessential examples of the devastation that we can see due to a lack of biodiversity is in the banana population. In fact, the Cavendish banana, genetically identical, and the Fusarium fungal pathogen literally spread like wildfire through that population, and there's no immunity, right? There's nothing, no part of the population that was resistant to that pathogen. So this causes a great deal of problems. So there are other questions that often come up around genetic engineering, and I know there's some of you even right now as I speak that are yelling at the screen and you're saying, Rachel, what about designer babies? Okay, so this is certainly one that people bring up a lot. That is, if we are going in and modifying genes, how long will it be until parents decide that they want to make their athletic, beautiful baby, genetically engineering it to have the characteristics that are desirable, quote unquote, right? Intelligence, quote unquote, whatever, remembering that we can't really define those things. And then it does sometimes have scary connotations. So I'm going to make note of some scientific elements that surround this, and I want to start by defining germline gene therapy. Germline gene therapy means that changes are made in the genes of the gametes or fertilized ova. So changes in genes of gametes or fertilized ova. Now, this is the kind of genetic engineering that would be needed if we were to treat, or if we were able to prevent, the fatal neurodegenerative disease called Tay-Sachs disease. We would have to be able to change that at the germ line. And of course, I think that all of us can absolutely 100% agree that it would be fantastic if we could prevent Tay-Sachs disease. But then one of the issues that surrounds germline gene therapy is the questions that we ask about what constitutes a disease. So I'm just going to make a tiny little note here that says, what is a disease? What is / who defines disease, right? And I think this is where Nick or Nicholas, I'll have to meet you before I decide exactly what I prefer. This is where this can become a human rights issue. So Nick expressed some interest in that, right? And this is where I believe we may run into issues, right? Because while we can all agree that Tay-Sachs disease is a disease, I think that this is much less clear when we ask about something like deafness. And those of you who have and love someone who's deaf will recognize that that person most likely identifies strongly with the individuality that they have as part of that very important population or group. Those of us in the queer culture, right, we don't want to be defined as diseased. And this is the question where we start to ask about what kinds of conditions, quote unquote, are we going to want to modify genetically at the germ line. So I think these are very worthy of discussion. So whereas I worry less about designer babies, I worry more about some of these, I think, poignant questions. I just want to make a quick differentiation between germline gene therapy and somatic cell gene therapy. Somatic cell gene therapy would be if we were going to be making changes to the already adult tissue. So if we were to define that, we might just say treats only the cells of the affected organ. This would be like if we, so only the cells of the affected organ. This would be like if we wanted to treat cystic fibrosis. Remember, that's just a deficiency in a particular transporter. It's a particular chloride ion transporter within the lungs. If we were to try to give that person's adult tissues the ability to suddenly make that transporter, that would be somatic cell gene therapy, only the cells of the affected tissue. And I was just thinking that probably, it's Maddie, right? It gets pretty jazzed about cardiovascular things. That was probably pretty interesting to you. So these are some of the questions. We can make a much larger list of this. We might even just add on to this. I think maybe let's put it up here after lack of biodiversity. Let's put the question of selective pressure. I almost believe that this may be the most important question facing us, period. So when we transform plants and create genetically modified organisms, so BT corn, right, is a good example of a genetically modified organism, or a GMO. When we have a GMO that produces its own pesticide and it's constantly exerting a pressure on the insect population, we can in fact select for the insects that are resistant to that toxin. So with time, we will simply see that the insect population, through selective pressure, will become resistant, right? So it's not that we're making these organisms resistant, that we're putting a constant selective pressure on them. And so the small percentage of the population that was already resistant, that is the population that thrives and becomes dominant within the larger population. Selective pressure, incredibly important question that we need to ask regarding genetic engineering. We could add also onto this the idea of lateral gene transfer. Maybe the idea that genes could be transferred, lateral genes transfer / pollination, like whether we're talking about a horizontal event or a breeding, right? If we talk about those plants that have the capability of resisting herbicides, and that those might pollinate weeds, we suddenly may have a population of weeds that are resistant to herbicides. And this is another consideration that we should put out there. And of course, lastly, I might just make note, and this is something that is going to be very interesting to Sammy. Sammy mentioned this. Your future maybe lies in looking at bioterrorism, right? So this is something that interests you. So this is the idea of purposefully genetically engineering microorganisms to be able to cause disease. So genetic engineering, can I just say GE? I know that sounds silly, but GE for bioterrorism. This is, again, a very partial list of something that I just want to plant the seed for your further thought. Let's leave this behind at this point, and I want to look at a subfacet of microbiology that is going to be of particular interest to Courtney and Trey. Both of you expressed a lot of interest in medical applications of microbiology, and let me tell you, microbiology has every link with medicine. Many of my students in the past have told me that this is the biggest no-brainer question that I ever ask. That's probably true. So which one of the following killed the most Americans? World War I, World War II, the Korean War and the Vietnam War combined, or was it the flu between 1918 and 1919? That is the 1919 Spanish influenza. We know the answer here, right? It's D, but you may not have realized that the 1919 influenza killed more than A, B, and C combined. World War I, World War II, Vietnam War, and the Korean War all together. So this is crazy. Between 20 and 40 million people are estimated to have died from this influenza. 500 or 500,000 just to American deaths alone, which is crazy when you consider that only around 116,000 Americans were killed in the World War I. So it's very, very different, right? And of course, we were in the throes of World War I in 1918 and 1919, and so thinking about that as a backdrop for World War I is pretty crazy, pretty epic to think about. So let's look at the historical connotations of medical microbiology and talk a little bit about it at a definition level: the study of disease-causing or pathogenic microorganisms. Pathogenic microorganisms mean those microorganisms that cause disease. We often say these are virulent microorganisms, and in fact, I'm going to jot down that term, virulent. Virulent microorganisms are those microorganisms that have the ability to make factors that cause disease symptoms. They are pathogenic. So if we were to turn back time and we were to look at the beginning of the 20th century, in fact, if you look at the Centers for Disease Control website, in fact, I suggest you guys should all go there if you have not already, cdcs.gov. And check this out, 1906, 3 of the 4 leading causes of death were infectious disease. One of them was pneumonia, one of them was tuberculosis, and the third was diarrheal disease. So literally, dehydration is the cause of death in diarrheal disease caused by things like, for example, cholera. So this was very much on the forefront in 1906, as was true throughout history. In fact, from 1346 to 1350, 25 million people died of the bubonic plague. In fact, maybe some of you know that the children's song, Ring Around the Rosie, actually is talking about the bubonic plague, the Black Death, right? We all fall down. So this picture is a picture that I took when I was bike touring in England. We were in a tiny town of Selby, and it's an entire mass burial ground for those who were killed by cholera. The cholera epidemic here killed over 100 people in a very tiny town, and so they were eventually able to put in Clean Water Acts that were allowing them to get water that wasn't contaminated with fecal matter, and that helped to prevent cholera. This is another picture that I took while bike touring. I was up in York when I took this one. This is the spine of someone who had systemic tuberculosis. So remember movies like Tombstone, where Doc Holliday was dying from consumption, or Moulin Rouge, where Nicole Kidman is coughing up blood. This is consumption or tuberculosis. It looks like you are all about ready to get to meet my cat. She's about to jump onto my lap. Her name is Gabby. So as she comes into the picture, you will know who she is. So with all this background in history, with regards to microbial infectious disease, it's easy to want to say, oh, we don't worry about any of that now. But I bet Trey and Courtney are thinking a little bit differently, because although we sometimes consider many diseases to be eradicated, which those of you who get to know me well will know that I kind of think of eradication as a cuss word, and we could talk about that more later. But although we now can say that infectious disease definitely does not top the charts of what causes death, we can say that we are getting to understand more and more how microorganisms may be behind some of those diseases that are causing death in many cases. We also are understanding that there are other new viruses and viruses on the rise, ranging from things like West Nile virus, to things like Lyme disease that are vector-borne diseases, yellow fever. Time magazine did a fantastic entire version dedicated to one issue dedicated entirely to infectious disease. In our discussion, I hope that many of you will decide to take on the discussion of Fevered, the optional book. And in that book, we'll be talking a lot about how changes in climate are leading to increases in vector-borne diseases, those diseases that are transmitted, for example, on the backs of mosquitoes, so to speak, that can now be the vectors, the mosquitoes, can get to regions and live happily in regions that used to be too cold for them prior to climate change. So this is something to definitely dork out about. DJ, you're going to want to be a part of that discussion, I have no doubt, because we are going to super nerd out. So because of international travel, so even as I'm talking right now, as you're listening, I bet I am currently on a plane to Slovakia, spending my morning here in the States and spending my evening probably already in Slovakia. So over a very short period of time, I'll be in many different locations. And if I was carrying an infectious disease, of course we know that would travel just as rapidly as I did. So we mentioned already old diseases making a resurgence, no more as much as vector-borne diseases, but also we're beginning to understand that the microbial world plays a role in diseases that were once thought to be only stress-related. For example, peptic ulcers, once thought to be caused by stress, we now understand are associated with a bacterium called Helicobacter pylori. However, we also understand now that in low concentrations, Helicobacter pylori can be protective against acid reflux, but in high concentrations, it can cause peptic ulcers. So this is crazy because we need to turn our attention to the balance in our microbiota to understand the ways that the bacteria can both help and harm our health. That balance is key. So lastly, I already drew your attention to this idea of antibiotic resistance, but I would argue that antibiotic resistance is one of the primary things that we face at this time. In fact, just this morning on my Twitter feed, which if you haven't found me on Twitter, it's The Virtual Edge, and I'll try to tweet out, I was reading an article just today about antibiotic resistance and in Staphylococcus aureus, and I was reading about the fact that sometimes co-prescribing vancomycin to treat a staphylococcus, an antibiotic-resistant Staphylococcus aureus infection. You guys have heard of MRSA, right? Methicillin-resistant Staphylococcus aureus. Some of you may have had experiences with it. Sometimes using vancomycin to try to treat an infection of MRSA, while simultaneously using another antibiotic to treat typical secondary infections of gram-negatives, may cause the vancomycin not to be effective on the MRSA. So there's a lot of sexy-minded dialogue to be had about antibiotic resistance. Maybe something you want to consider doing your poster on. Man, I think within the last 2 minutes I have presented about 500 different possible poster topics. So many trendy things in micro.